Hello, everyone. My name is Danielle Azariti. I'll be moderating our career panel today. So we're just going to give all our attendees a moment to join. And this is week 2 of the ClinGen Clinical Genomics Career Panel with our clinical laboratory professionals today. So welcome.
Thank you.
Okay, it looks like our number of attendees is staying pretty constant. So we will get started. I'm just going to start with some introductions. So welcome everyone again. This is a 5-week panel that ClinGen has been putting on. We're glad you found us. We are in week 2 with our clinical laboratory career opportunities. That's the panel you'll be seeing today. We do have 3 more weeks. So this time for the next 3 weeks, there'll be other opportunities to learn about other careers in the field. And also all the recordings will be made available online if there's anything you miss, or if you know someone else who may be interested in hearing one of these sessions, please feel free to spread the word. And these will be available on our website, and we'll flash them up at the end again as well. And to give you some orientation to Zoom over the course of the webinar, all of our attendees, you are on mute, but you do have the ability to either chat or enter a question and answer in the Q&A box. We encourage you, any questions for our panelists, please do use that Q&A function. We'll be collecting them over the course of the discussion and asking our panelists as they come in. So please take advantage of that. And you will also notice if you see a question that you would like answered, you do have the ability to upvote it. So give it a boost to bring it to the top. So feel free to use that as well. Now we'd like to get to know you, our attendees, so the panelists know who their audience is. So we just have 2 quick poll questions for you. Hopefully one just popped up on your screen. As of fall 2021, are you a student? So please let us know. I'll give you a moment and then we'll be showing everyone those answers. Okay, so the majority of our attendees are undergraduate students, but we have a number of recent graduates, PhD students, master's students. So thank you. You are all welcome here. And next question. How familiar are you with clinical laboratory careers? With the goal being that by the end, you'll all be very familiar. Okay, so the majority are somewhat familiar. You're here to learn more, then you are in the right place. So thank you for your participation there. Before we meet our panelists, we are going to have a quick presentation by one of our panelists. Marina Destefano is going to give you a quick overview of a clinical laboratory setting and different careers that you'll be hearing about today. So with that, I will turn it over to Marina.
Great, thanks, Danielle. So these are just some really generic overview slides. I personally did a lot of my training in a research laboratory. So coming to a clinical laboratory, it was a bit of a shock in the beginning. So I just wanted to give a little bit of a broad overview about clinical labs. And there are many professions or many personnel within a clinical lab. You'll hear about the career paths of some of them today. So I don't necessarily have all of the training steps for some of the personnel, just because people can take really different paths within a clinical lab, but I'm hoping that the panelists can really explain to you their experience and the training that they've had. So what is a clinical lab? In general, a clinical lab is a lab that performs testing mostly on humans. You could have a veterinary lab, I guess, and returns results that are added to a patient's medical record. So as of 2003, all clinical labs that perform testing in the US have regulations, and these are generally by the Centers for Medicare and Medicaid Services, CMS. So different labs are required to maintain different certifications. Many of the certifications that you'll hear within the genetics sphere fall under CLIA. So this stands for Clinical Laboratory Improvement Amendments. And this really is to promote consistency, quality, and transparency in laboratory testing. So at this point, when I looked at maybe last week, CLIA currently covers approximately 260,000 laboratories or tests. And this is often grouped by complexity of tests: high, moderate, and waived. And many of the genetics tests that we think about, exome genome sequencing, for example, are high complexity tests that are regulated by CLIA. So some examples of clinical labs, and many of these will work together or overlap. You could have a microbiology lab. I know with the pandemic, microbiology labs really had to throw up a lot of COVID testing. This could often overlap with a molecular genetics lab, for example. That's another example of a lab. Cytogenetics lab, particularly with the merging of a clinical laboratory fellowship, which you'll hear about from some of the panelists. Molecular genetics labs and cytogenetics labs have often worked together now or combined, and potentially pathology labs. Those are all examples of clinical labs. Next slide. So just to give you some comparisons from a clinical versus a research lab, and to step back for a second, these labs are not mutually exclusive. And so more so recently, the boundaries between the two have kind of blurred a little bit, but research studies often take place in CLIA labs. So oftentimes when we think about CLIA, it could potentially be the test, it could be the bioinformatics pipeline. It isn't necessarily the physical space where the lab is that is a clinical lab versus not. So, but I just wanted to give you a little bit of a comparison about maybe purely clinical labs versus research labs, or clinical tests versus research. So for clinical, this is often indication-based testing, doesn't necessarily always have to be the case. Many labs will return results to doctors and patients, putting this in the electronic health record or the EHR. And there are certain standards that are required for maintaining accreditation, kind of like we talked about for CLIA right now. Certain certifications or training are required for certain lab roles. Maybe board exams are required, different certifications for different personnel, and you'll hear about some of that today in training. And then there are very structured procedures, often for validation, for new methods, machines, programs, bioinformatics pipelines. They really all have to be validated to be able to be used within the process and to return results to patients. So up-to-date procedures and SOPs that are date stamped are required. Sample documentation is very thorough. Often there's a barcoding system. There are multiple pieces of identifiers that are used for each sample, because it's really goal not to have sample swaps when you're returning results to patients. And so oftentimes LIMS systems or laboratory information management systems are used in clinical labs. For research labs, oftentimes they're discovery focused. Maybe this can be disease research, this can be basic science. There could be a range of different organisms that are studied. So you have human research, of course, maybe gene discovery, cell culture, model organisms. And research labs aren't just the Wild West. They do require their own ethics boards too. Oftentimes, so IACUC is if you have vertebrate animals, oftentimes mice, rats, primates. The IRB is for human studies, and really detailed and consistent protocols need to be written for these ethics boards, and they need to be reviewed and these protocols be followed. So for particular experiments, maybe that don't necessarily involve human subjects, maybe cell culture experiments, these experiments and protocols can be fluidly changed to maximize their optimization and to optimize them over time so that they can then be published. So really less documentation, formal documentation is required oftentimes when you're trying to discover new assays. And these labs are often funded by external grants, maybe from the NIH is a huge funding source, or other research foundations. And it really depends on the protocols with your ethics boards, but result return is contingent upon the study design. And in general, unless you couple a clinical lab result with a research result, it generally does not enter a patient's medical record. But of course, this is really dependent upon each study. Next slide. So I just want to talk about for a second wet lab versus dry lab roles. And personnel may have roles that span both the wet and the dry side of the lab, but just to give you a sense before you hear from each of the different panelists. So when we say the word wet lab, we often refer to those personnel who are physically in the lab. So these could be people who work with sample receipt and accessioning, who perform the tests physically, do quality control of the reagents, the physical machinery, who do skill assessment for those individuals who are being trained to run the assays, and then maybe equipment testing and maintenance. When we talk about dry lab, this is often personnel who participate in the process outside of the lab. And for genetic testing, this really can be the variant science team or the bioinformatics team. So this could be interpreting the results outside of the lab, bioinformatics and software design to support the testing and management. Quality control also needs to be done for the dry lab side. So what interpretation programs are you using? How are you filtering your variants that you get off of your tests? Quality control, skill assessment also needs to be done for those individuals who are within the dry lab. And then also those individuals, we have many laboratory directors with us on the panel today who draft reports, or even the variant science team, and work to return those results. And there are even laboratory genetic counselors who you probably heard from last week in the genetic counselor panel, who consult with patients or potentially physicians with test ordering or result return. Next slide. I actually borrowed this from the genetic counseling slide from Jules last week, but there are a range of different career possibilities. Laboratory professionals work in a variety of settings. There are clinical labs at universities for ease of use that oftentimes pathology labs or cancer labs or something that you need a result very, very quickly. You'll often have a university lab or a hospital lab. There are labs at the government. We have individuals who are working in the government. There are labs in industry. So there really is a range of different settings and professional opportunities for laboratory individuals. And I just left these specialty areas here because any one of these specialty areas that you see on this circle often has associated clinical labs that do that particular type of testing. And depending on the lab, they may have a particular specialty of a certain subset of tests that they would do. So there really is a lot of flexibility for career possibilities within clinical labs. Next slide. And here are some just additional training resources. So there are different training options for geneticists. I put the American Board of Medical Genetics and Genomics and the training options there. These are often for individuals with advanced degrees of MDs, PhDs, or MD PhDs. There's the National Society for Genetic Counselors if you're interested in laboratory genetic counseling. There's the American College of Medical Genetics and Genomics, and also the Association for Molecular Pathology. And there are many other wonderful resources. This is just to name a few. So I think that's all the overview slides that I have about a clinical lab.
Thank you so much, Marina. That was a great overview. So I'm now going to introduce you to our panelists. So we have 7 panelists joining us today from a range of different laboratories who have different backgrounds and specialties. So to get us started, I'm going to introduce each of them, and I'll ask that they just give a brief introduction about themselves, where they work, and what took them on that path. And then we'll get into the questions. So feel free to start putting those questions in the chat for us to get to after the introductions. And with that, I'm just going to call our panelists out one by one. And we'll start with Rashid. Rashid, please introduce yourself. Thanks for joining.
Hello, thanks everybody. Thank you for inviting me. So my name is Rashid Karam. I am currently the Director of Clinical and Translational Research at Embryo Genetics. I have been with Embryo Genetics since 2014, and I am an MD by training, and I did a PhD subsequently, a PhD in oncogenetics.
Thank you. And next up, we have Daniel.
Hi, my name is Daniel Pineda. I am the medical director of hereditary cancer in VITE, and now transitioning is the director of medical affairs for Latin America. Like Rashid, I'm MD. MD. I trained as an MD in Colombia, moved to the US, and pursue a lab career instead of a clinical career. And I can tell you later why I made that decision and how that happened. And yeah, I'm happy to to open the book of my life to you guys and tell you all about it.
We will take you up on that. Thank you. And next up, we have Marcia. Thank you for joining us.
Hi, my name is Marcia Pasquale, and I have a PhD degree in pharmaceutical chemistry. I did my graduate studies in Italy, then I moved to the United States, and I pursued a fellowship in clinical biochemical genetics. Currently, I have two hats. I'm a professor of pathology at the University of Utah, but I'm also the section chief and medical director for biochemical genetics at ARUP laboratories. So I have both academic and clinical responsibility.
Thank you for joining. And next up, Raj.
Yeah, hi everyone. So I'm Raj Kosh, and I did my PhD in genetics, classical genetics. We did my postdoc in more of a quantitative statistical genetics, and then I basically wanted to switch fields. I did it in model organisms, then switch my fields to kind of clinical genetics. That's why I was a staff scientist with ClinGen for a few years. That helped me get into a fellowship, which is the Laboratory Genetics and Genomics Fellowship, which is we were the first batch of that kind of fellowship. And then after board certification, I joined the NIH as a clinical molecular geneticist. And we are working primarily out of NIAID. And you can ask me any questions about this whole career trajectory later. I'll stop there. Yeah.
Excellent. Thank you. And Carolyn.
Hi, I apologize that no one can see me, but I have worked in clinical cytogenetics for almost 20 years now here at the University of North Carolina at the hospital. And most of my education is in Canada, but I did do a master's degree at the University of North Carolina in molecular diagnostics. So I am dual certified for cytogenetics and molecular genetics. But I mostly practice genetics. Yeah. So I'm a senior cytogenetics technologist here.
Wonderful, thank you. And Urjit.
Sure. Hey, everyone. So yeah, my name's Urjit, and I started working in the wet lab for Quest, running next-gen sequencing. And then around 2013 is when I decided to go to school and finish my master's in biotech, and then switched over to doing variant science work around 2018. So I'm fairly new. It's been a few years, but at the moment I'm the lead variant scientist, focusing more on the CNVs and the larger structural variants at Quest Diagnostics.
Wonderful, thank you. And finally, Marina.
Hi, everyone. I'm Marina Destefano. I'm a board-eligible clinical molecular geneticist taking my boards this summer. I am an assistant professor at Geisinger and also part of the ClinGen staff. So I work very closely with the ClinGen staff, and I have since 2016. And I also work on the MyCode project at Geisinger, which is kind of their community health initiative, and it's a screening project. And I have a PhD.
Great, well, thank you all. We're excited to get into this discussion. We have a few questions coming into the chat, so I'm just gonna pose them to you, and feel free if it's a question that you're ready to answer, go right ahead. If several people have answered the question, we can always move on to the next. So we'll see where this goes. So let's start off with the classic. What made you decide this career path? Anyone in particular want to get started?
I can start. So we start open chapter 1. So around the time I was finishing my my MD training. So there's here in Colombia, and I'm in Colombia this month. So here in Colombia, training is sort of different than in the US. It's a 6-year program. The last year is the internship. In the internship, you're basically you're basically seeing patients. The attendings are very peripherally involved, and you're basically handling the patients and the services. And I had been at NIH already a couple of times during my MD training, and I just fell in love with NIH and the lab work, and being able to score new genes for that time was developmental disorders. I was also involved in ADHD, which is a complex disease and common as well. And also the stress of the internship. I'm like, I will have a heart attack when I'm 40. And I don't think I will be able to learn at the same scale as the NIH at that time. I wouldn't be able to learn and how to discover genes here in Colombia. The resources, at that time were very limited. So I took Max Munca's offer on a postdoc, and I decided to move to NIH right after I graduated from my school. And then it went on. Before I actually joined Max's lab as a postdoc, I had been begging him, please let me do the clinical molecular training program. That's what I would like to do. That's my passion. And he was like, no, let's wait until you graduate. Come as a postdoc and we'll see. And finally, after years of postdoc, he came to me and said, okay, Danny, it's time for applications. Are you still interested? And I'm like, yes, I'm going to apply. So I applied to the NIH program, which is a bit different than most of the training programs in molecular genetics across the country, because at NIH there's no funding for the fellows, so you basically have to enter to a postdoc in a lab. So you're doing both your postdoc and your clinical training at the same time. So I did that, graduated. And part of my work as a postdoc was doing characterization of patients with holoprosencephaly by RACGH. And all that work, because we didn't have a scanner in campus, I had done all my training at GeneDx and used GeneDx's scanners and the ISCA array at that time. This is back in, 2009. When the ISCA was studying and had put this whole genome arrays that were targeted at the exome level. And there were fantastic arrays. So I had been doing those arrays on my holoprosencephaly patients at GeneDx and got very familiar with the analysis and how to interpret CMVs, etc. And got very close to Swarup, Aradia, and Sherry Bale. And at some points where I wanted to move, to do some epilepsy work and not the array work that he had been doing. So he's like, okay, Danny, would you take this position so I can move and do some other stuff? And I end up joining GeneDx after I graduated from the fellowship. And then it kept progressing from there. It was, to me, it was just love for the lab work and wanted to discover new things at a scale that wasn't possible here in Colombia, and honestly not wanting to do 24/7 hospital work.
All right, thank you for that. Did anyone take a different path that brought them to the career they're in today?
I can tell you my story. Okay. So my background and my training has been mainly in analytical chemistry and, believe it or not, physical chemistry. So, but I've always been interested in applying this knowledge to biological system. So this, when I moved to the United States, I was exposed to a biochemical genetics lab. And what I loved about that is that again, I could use my analytical chemistry skills, but I also was able to solve puzzles and put together the information that that you are getting from the different tests that you run in a biochemical genetics lab to understand what is behind a patient's symptoms, and you can help. And especially when you're dealing with metabolic disorder and you make a diagnosis, the beauty is many of these disorders have a therapy, not yet a cure, but a therapy, and you can improve the life and the outcome of this patient in a tremendous way. I mean, it's very, very rewarding. So to me, this is what is exciting. The idea of being able to do screening in the neonatal period. I've worked with newborn screening for many, many years now, as you can tell from my white hair. But, you know, doing the screening, coming up with new methodology or way to identify new conditions, seeing the advances in the therapy. Every day in our field, there is probably a new therapy, a new clinical trial. And you can follow. The other thing that's exciting for me is that you can follow these patients from birth and throughout their whole life. And you see them, they start school, they have a family, they are really doing living a pretty normal life in many cases. And the fact that every day is a new challenge, is different. You're looking at reports when I am reviewing reports, each report is different than the other, and you need to integrate the clinical information with your laboratory information. We work very closely with the clinical team. So this is what got me into this field, and I'm still hooked on it. I still, after 30 years, I still love what I'm doing.
Good to hear. Well, we have several questions coming in about the degrees that you all have, and what how you chose those, or what options are available to you depending on the degree you have. So for those who weren't able to cover how they got into the career path, I think you can probably work this into other responses as well. So we'd like to take that. So what can you do with a PhD versus a master's versus a bachelor's? You know, most of you have advanced degrees, but have had several different jobs over the years. So maybe you could talk about some of the differences there.
I think that question kind of fits with basically the way I got into this field. So I can cover a few other questions. I see it's there, what can a PhD in genetics do in a research genomic lab? So it will cover a few questions, I can say. So I'll just give you that I had no idea when I was doing my PhD in genetics that I would go into clinical genetics. And I just love genetics, that's all it was. Then I did my postdoc in statistical genetics and quantitative genetics. That was just for love of genetics. It was all in worms. So by the time I finished my postdoc, I mean, the sequencing, this was like around 2011, 12, the sequencing, this was before, if you guys are familiar, this was before the ACMG guidelines for variant interpretation. So at that time, I thought that, okay, the sequencing in human genome sequencing price has been dropping so fast and it's becoming so low. So maybe I would like to do something applied using my genetics knowledge so far. So at that, and coming back to the question, and that time I applied directly from my postdoc to several programs for molecular genetics fellowship. And I didn't get into any one of them, but they were very nice to give me advice and reply that you need to have some experience in molecular genetics. I mean, the CVs are great, but you have no experience in human clinical genetics. clinical diagnostics. So you need a little bit of that, at least human genetics PhD or clinical diagnostic experience to get into these fellowship programs. So then I kind of stopped applying, and then I looked for opportunity. And that time, ClinGen was basically, there was, actually I got this from Twitter. Dr. Sharon Plon, she tweeted that someone, they're looking for staff scientists. And I joined, I just applied and joined. So there was a big leap coming from a warm background, going into clinical genetics in a completely different job. But that was the most rewarding experience. I learned a lot, and it helped me get into a fellowship program, which is the LGG, and eventually got into where I am, the centralized sequencing program at NIAID, which basically where you can see patients who you're sequencing and return the results, as well as you can do research as much as you want. There's no restrictions for doing research with the data you have. So the thing in this, basically the bottom line here is that I think from my personal experience, it's hard to get into the LGG fellowships, especially now, given it's become more and more competitive, without absolutely no human genetics background or any experience, a little bit of experience in clinical diagnostics. And the other thing take home from here is, and the experience that you get, it's later when you're kind of a lab director or you're signing out patients, and you also want to do research, like the environment in NIH is always helped to have some quantitative skills like programming, and that way you will have access to a lot of data, and you can analyze and you can come up with the research questions if you're interested in that part as well. So I hopefully cover a few questions.
Good, thank you, thank you. And did anyone on the panel hold a position when they had a bachelor's degree, for example, before going on? Yes.
Yeah, so I was just gonna say I might be the odd one out here without the PhD. So I started as a bachelor. I didn't have even a master's when I started at Quest, and I was basically, a medical technologist, but just from like, enjoying the work and like showing more interest in like, the theory of it, I was encouraged to go back to school and like get a further degree. And so choosing between a PhD where I would have to leave work and do it like as a full-time thing, the other option was to do my master's part-time and then keep my full-time job at Quest. So I actually decided to go that route just so I can have money coming in. And it worked out in the sense that it's not required, at least at Quest, it's not required that you have a PhD to do the work in the lab, as well as doing variant science work. You do need the PhD if you're going to be a director, and if you want to be boarded, all that stuff does require a PhD, but not necessarily for scoring variants, assessing variants, and writing reports. All of that I was doing even without the master's. But the master's I completed recently, and it kind of did help in holding a higher position within the variant scientist group and potentially be manager at some point.
Excellent, thank you. Any other perspectives there about the different opportunities or a reason you pursued a degree for an opportunity?
Hi, it was Carolyn again.
Oh, hi, Carolyn. No, I'll do Carolyn, then Marina.
Sorry. So I just started my career with a bachelor's degree as well, and I did like a clinical genetics program in Toronto, and there's similar programs in the United States, I think, offered through MD Anderson and UConn as well. And then I went on and did a master's like probably 15 years into my career. And UNC offers an online program, and some teaching hospitals also have like tuition reimbursement, which is what I did. So because I continued to work here, I was able to get most of my master's paid for, and most of it was online, which made it convenient to still work at the same time. But to get in as a technologist to cytogenetics, you just need a bachelor's degree with a couple genetics classes. If that helps.
Thank you. And it sounds like from what we're hearing, those bachelor's level positions can be standalone careers or opportunities to learn more about the degree you'd like to pursue. So Marina, I think you had a perspective as well.
I was just going to echo what Raj said, because I know this is one of the common questions that I get the most about the laboratory fellowship, that I think often now it is very difficult unless you have a very targeted PhD to go straight from a PhD into a clinical fellowship. I did the same thing where I applied straight out of my PhD to clinical fellowships. And I just want to point out that oftentimes it doesn't cost you very much to apply. So maybe it's worth it to explore. And that actually led me to my opportunity. So I applied to a bunch of programs. Everyone was very nice. They, just like Raj said, that you have good qualifications, but not necessarily maybe the exact ones. But that's actually how I got my postdoc, that Heidi Rehm, who was one of the directors in the molecular fellowship at Harvard, emailed me and said, "You're rejected to this fellowship, but I would love to recruit you as a postdoc." So I actually spent a year as a variant scientist in her lab and a postdoc, and then transitioned into the fellowship. So even if you feel like you don't necessarily have the appropriate qualifications straight out of a PhD, sometimes it doesn't hurt to apply, because people are really looking for talented staff they can recognize when someone has the potential and maybe give you those opportunities that would help you to then get into the fellowship. And I started working with ClinGen in 2016 when I did my postdoc, and I've worked with them ever since. I'm still very closely connected with Heidi, and all of my opportunities following have really been through those connections. So it's not super likely that you would get into a fellowship right after your PhD, or maybe even a short postdoc if it's not related, but it doesn't hurt to try. I would say they really value experience now in molecular genetics, and now that the specialties have merged, laboratory genetics being molecular genetics and cytogenetics combined, that they even more so like to see experience across both. So if you can find a lab that maybe has cytogenetics and molecular genetics and get a bit of experience, that's even better. But I think that they do require some experience now to get into these fellowship programs, because they're fully funded programs, and most people can only take one person a year, maybe up to 5. I know AROP, I think, has a decently sized program. It's one of the larger programs. New York also has some larger programs, but it's relatively competitive. And a lot of times they are getting internal applications from postdocs or variant scientists who are always work there, who already work there. So just to kind of follow up a little bit on that, because that's a question that I get pretty often.
Thank you. This is great. So it sounds like networking, getting your name out there, meeting people in the field, Twitter, these are all opportunities to learn more and make connections. One question we got that I think this panel is poised to answer and address is, what are some of the major differences between academic and industry lab settings? I know we have representatives from both and from NIH. So if anyone would like to speak to things they've noticed or what they like or dislike about the current field that they're in.
I can address this one because before I joined Ambry, I did my postdoc at UCSD, and it was a traditional academic postdoc in molecular biology. So it was really like you have a specific project and you really work on that project. A lot of times projects in academia, it's more about your individual contribution. There is a lot of networking, a lot of connection, but that's your project and you're really focusing on it. And usually those academic projects, can be multiple years long, right? Like my postdoc, for example, it took almost 5 years. And when I moved to industry, I think one major difference, it's like things are much more fast-paced. You in 1 year, in a couple of years, you can be working multiple different projects. It's a lot of teamwork, right? It's rarely you work on a project by yourself. Usually there are multiple people involved in a project. So you need to be very good with teamwork and fast pace. A project rarely lasts more than a year or two in industry, even research projects. So I think the speed that things happen in the industry is very different. And also, but one thing that I was surprised that I didn't find as different, I thought that necessarily things that industry would be very focused on on commercial, but no, actually in industry or in commercial labs or diagnostic labs, you can find questions that are very pertinent as well as you would find in an academic setting. So it's with regards to the questions you can ask, I think they both are comparable. It's more like the infrastructure and the pace of the work that I found to be very different between industry and academia.
Excellent, thank you.
Now, if I may, I can add a little bit through that. So there's this misconception that once you move, and people who really like to do research and like to drive their careers that way, there's a misconception that the industry does not do research, that these labs do not do research, and I don't think that's true. Maybe the scope and the focus is a little bit different, but I think the clinical laboratories still, like any other hospital, help drive patient care and help drive that research that actually improve patient care and at the end the health outcomes of these patients. So it is important to sort of like burst that misconception of industry does not do research at all.
And I realize I may have introduced the question as industry versus research, which I did not intend. So thank you for pointing that out, because I know on the ClinGen side, the research we've seen on both academic and industry sides, really, I think the two couldn't interact without the other. So it's great to have all sides represented.
Okay, and if I can add just a couple of words, my perspective is a little bit different because I'm in a big reference lab, but it's an academic lab. It's fully owned by the University of Utah, so it's in between. So I think the big difference is really in the infrastructure, like Rashid, I think, said earlier. I trained and worked at Emory University for 15 years before moving here. And in a smaller laboratory, you have to do a lot of things yourself. You're doing pretty much everything from testing to reporting to marketing to billing, everything. In a larger institution, you have an infrastructure that is helping, and you have different departments that take care of different things. But again, I think everywhere the commonality is we're here to improve patient care.
Thank you. And another good question. So would anyone like to describe what their typical day looks like in the current job that you do now?
Sure, I can take a stab at this. So in the clinical setting, as a variant scientist, you're, for the most part, you're responsible for assessing variants that directors send and writing up the report. So I would say that's probably like 50% of the work. And then the other 50% is like, process improvements. Recently, we updated our SOP based on the ClinGen guidelines that came out at the end of 2019. So I was heavily involved in working with the directors to draft up an updated SOP. So then training others once that gets approved by the committee. So yeah, just it's 50% you're doing like day-to-day scoring variants, and then 50% you're doing peripheral work that sort of helps you make the process more efficient and improve it. So
thank you. Any other day in the life stories you'd like to add?
I can answer that again. As I said earlier, every day is a little bit different, but it starts with a walk to the lab and looking at the lab, understanding what, if there are any issue, any problem, any crisis that need to be addressed immediately, and meet with the supervisor of the lab and strategize if there are some problems, and then about, you know, a good, I would say 20% of my time is going to be administration and administrative responsibility, like quality control reviews, those SOP review. About 50% clinical service, doing looking at report, patient report, consultation with physician, and then the rest is teaching. There is a lot of teaching going on. We have 2 fellows, and we have genetic counselor students. We have other fellow in other specialty that who rotate in our lab. So the rest is really teaching and research.
Thank you. And does anyone else have teaching responsibilities? We did get a question about how people balance being both a professor and having a clinical lab role.
I do a lot of teaching and a lot of training. My role is maybe a little bit non-traditional. I direct the biocuration course. So for ClinGen, we do a lot of expert level curation, and many of these individuals are very busy and volunteers. And so we have a whole staff of biocurators who are usually research assistants straight out of college. Often it's a gap year position. Many of them have gone to medical school or master's in public health, nursing school. So I do some really intense training of those individuals to get them going to be able to pass them off to all of the expert panels. I sit on about 15 or so expert panels myself to also provide guidance. So I do a ton of teaching of molecular genetics, variant science, of those individuals. And then also within the Harvard system, I've done a bunch of teaching, exome genome analysis to residents, to fellows, to my fellow clinical fellows, and I'm now starting to teach medical students at Geisinger. So sometimes it can be a little bit difficult to balance. I think the goals are maybe to group your teaching as much as possible instead of having a bunch of separate teachings on similar things, maybe to try to teach as many people at one time, have kind of standard decks of slides that you use, obviously update them if appropriate, and just try not to stretch yourself too thin. You don't have to necessarily accept every teaching opportunity that people ask you to do. I've turned down a couple workshops this summer because we just hired 3 new research assistants, and I'm intensively training all 3 simultaneously. But that is a big joy of mine, is teaching. And also it really helps you with your understanding. You don't realize until you start teaching people that you really have to know what you're talking about to be able to explain it to someone, particularly within molecular genetics, if they don't have that type of experience. So it's definitely been a great thing to help me study for boards too, is teaching people in molecular genetics as I go.
Great, thank you, Marina. And we're coming down to our last 10 minutes. We have two different questions that I think I can combine into one regarding opportunities outside the US. So this is both in terms of the way the questions are posed, someone with training outside the US pursuing one of the opportunities that's been described here in the US, but also for those of you who did your training in the US, what opportunities could you seek in Europe, for example? You know, how much is your training transferable to other countries, if anyone has perspectives on that?
I kind of have a little bit of perspective because I have been in 3 different continents pretty much. So I did my MD in Brazil, I'm Brazilian, and then I moved to Europe where I did my PhD. I did my PhD at University of Porto in Portugal in oncogenetics, studying CDH1. This is back in 2003, and just for you to like how the PhD actually carries over, right? I did my PhD in CDH1, and today I work with ClinGen. I'm the co-chair for the CDH1 expert panel. So, you know, like that work stays with you. And once I finished my PhD, I moved to the United States and I did my postdoc at UCSD. And I think that's one great advantage of a PhD. You can really easy travel and move across countries, across continents. It's like the knowledge that you acquire, it's something that it's very easy to translate, regardless of the country there you are. And then once you're there, once you show your passion, you show how much you know about a specific topic, if it's relevant, I'm sure the opportunities will appear we were rising, and that's how it worked for me, you know. And I'm quite sure that how it would work for most people that when they follow their passion.
Yeah, I can speak to certification, at least from co-fellows. Other countries, I can tell you, Canada will recognize the laboratory certification. So FACMG, Fellow of the American College of Medical Genetics, that we're all talking about for board certification. They also have their own, and some labs will require you to have an FACMG and a CCMG, which is the Canadian College of Medical Genetics. As far as, I think I have had some colleagues who've trained at Harvard and gone back to practice in Asia. We have some individuals who have gone back to Singapore, and they clinics there, and they act as clinical geneticists there. I know I have colleagues in Australia. They don't necessarily always come and do the American training programs because I think that it's maybe not recognized as formally there. I think that some of the American training programs are starting to be recognized in other countries. I think Canada is maybe the best direct translation, but I think if you have an FACMG, other countries will recognize that you've gotten a certain standard of training. It's just, you might have to take other exams in those other countries. I'm not 100% sure about that.
I would say that that varies country to country, particularly if your training is a clinical training. It will vary country to country. There will be some countries that will accept the credits and the credentials. Some will make you do some work on, some will actually make you start from scratch. So it varies from country to country.
Excellent. Thank you. Okay, 5 minutes left. Anyone wants to speak? I have one last question. If anyone wants to address, were there any obstacles to getting to the career that you have now that got in your way that either you didn't anticipate or you had to, you know, it took work to get over that? And if there's anything you'd like to speak to there, thinking about the students on the call who are facing careers ahead of them, any advice given your experiences overcoming obstacles? And if not, that's okay too.
Well, I think that the only advice that I can give a graduate or an undergraduate student who is thinking about this type of career is really to do some homework and get to know, if possible, visit a laboratory or shadow someone who is in this field to really understand if this is what they like to do. Because again, I think, I personally think it's a wonderful career. As I said, I love what I am doing, and but if you don't, if this is not what you really like, it might not be as exciting. So I think just get to do some homework and understand, is this the environment that you really want to be in? Because this is a heavily regulated environment. So from the technologist to the medical director point of view, there are very specific protocols that need to be followed. There is very little that's left to improvisation. You can modify things, you can improve things, but you need to follow a very rigorous path of validation. And again, it's not like in a research lab where the temperature of the experiment, well, it's not quite working at 37 degrees, let's bump it up to 38. That's not something that can be done. So it's a different environment. I think Marina said it was a shock going from research to the clinical lab. And I think that's the truth. So do some homework and make sure that this is really what you want.
And you can just add something quickly with regards to challenge too, like Marina and Raj mentioned that too. You will apply to many different positions, to many different jobs. You will get rejected, you get no's. it's okay, it's normal, it's part of the deal. I think just keep applying, keep trying, and eventually things will happen too. So everyone, I'm quite sure all of us got notes, you know, you not now, maybe this is not the opportunity, that's not the time, but over time, you know, if you're following your passion, things eventually happen.
Yeah, I will just echo Rashid that that's absolutely right. That's what you will basically face, lots of rejections. And one thing, just a practical thing, you may have to move for the fellowship. Like, you know, that's a big thing when you are, say, at least when after your graduate school. So you have to be prepared to move after your graduate school and postdoc. I mean, it's not certain that where you did your postdoc that you will get a fellowship right there or nearby. You may have to move different places around the country.
I was counting, I moved 18 times since my so just for you to, that's a challenge for sure. 18 times since like I got into med school. So.
Well, thank you all. I think we are right at the end of the hour. I appreciate all your perspectives so much. This has been absolutely, I found it informative, and here I am sitting at my career. So thank you all. I think we'll have, Jules may just have a last slide as to remind everyone that addition to the wonderful careers you heard about today in clinical laboratories, we have physician clinicians coming up next week, research development, other opportunities. You'll see there's a lot of crosstalk between all our panels. And finally, for those asking about opportunities for those at the bachelor level, we do have a number of professionals who have had different opportunities along the way to get into the careers they are now, such as genetic counseling assistant, biocurators. They'll be speaking on July 28th. So for those looking for your next step along the way, that could be an informative session as well. So thank you all for joining, attendees and participants. The recording will be available at clinicalgenome.org/careers from that and from past sessions as well. So thank you for attending.
All right. Thank you.
Thank you.
Bye-bye.
Thanks so much.